So good morning, good afternoon, good evening, wherever you are, and welcome to this WHO consultation on novel manufacturing technologies for monoclonal antibody production with a focus on prevention of infectious diseases for LMICs. This session is recorded, and we will share the recording by email with all who registered to this session. Please, during the webinar, don't hesitate to ask your questions at any time using the Q&A box that you will find at the bottom of the Zoom window. And in case you have any technical issue or you would need any assistance, please use the chat box, and I will assist you as much as I can. TechNet 21 is happy to host this session, and we hope you will enjoy every bit of it. Thank you very much, and Erin, the floor is yours.
Thank you, Alex. Hi, everyone. I realize that this is sort of an odd hour for some of you, being very early in the morning or late in the evening. So thank you for those of you who are joining outside of the normal working hours. For those of you who don't know me, I'm Erin Sparrow. I work in the Vaccine Research Unit in our Immunization Department at the World Health Organization. And we have an interest in monoclonal antibodies for the prevention of infectious diseases, which is the reason that we've organized this consultation. So the purpose of this meeting, or the purposes, are really to review the landscape of infectious disease monoclonal antibodies in clinical development, to discuss pathogens where the development of preventative monoclonal antibodies would address an unmet public health need, and to just really learn more about innovative technologies that may lower production costs and optimize product attributes. Because we know that these products are expensive products to produce, and we also know that when they're given in high volumes, they need to be administered intravenously, which is obviously not ideal for resource-constrained settings. So we really also want to sort of discuss how we can advance some of these innovative technologies. We'd also like to look at some of the product characteristics for use in low and middle income countries, including price and, of course, the route of administration, as well as others. And the expected outcomes of this consultation is really to increase awareness of the pipeline among key stakeholders. We've got about 100 registered attendees at this conference. And we want to discuss the feasibility of developing WHO preferred product characteristics for monoclonal antibody-based interventions, and I'll provide a little bit more information on that in subsequent slides. But as I said, one of the main purposes is to identify the challenges in advancing these novel production technologies so that we can advance affordable monoclonal antibody supply that is appropriate for low and middle income country use. And we will be summarising the findings from this meeting in an article that will be submitted to a peer-reviewed journal. So the agenda for today is I'll be providing an overview of monoclonal antibodies that are in development for prevention, as well as providing some more information on what WHO is doing in this area. My colleague Richard Ipsbrucker will present on some of the normative work that WHO is doing, and we'll hear from IAVI about efforts that they are undertaking to ensure affordable access to low and middle income countries to monoclonal antibodies. We'll also have a bit of an introduction to current and novel production technologies, and then we'll focus the rest of the day on disease-specific case studies. So with that, I move to the first presentation, which is from me, so that's easy. And so this first slide here actually shows the licensed monoclonal antibodies that are already on the market for prevention of infectious diseases. You'll see that there is a couple there for pre-exposure prophylaxis of COVID-19. There is one monoclonal antibody authorised for the prevention of recurrent C. difficile, two monoclonal antibodies authorised for emergency prophylaxis against anthrax. There are three products authorised for rabies post-exposure prophylaxis, And there's one product currently on the market for the prevention of RSV in very high-risk infants and high-risk young children. And what you'll notice there, I'm sorry if it's a little bit small for everybody, but you'll notice that the dose really differs by product, including the way that dose is administered. So some products are administered in intramuscularly, subcutaneously, but others require an intravenous infusion. And what you'll also see is that the cost of the products really vary quite a lot. This is just what we could find in the public domain on costs, and they may vary by country, and they may vary by whether or not they're sold to the public or private sector. But it's just to give you an idea of how different these costs can be. And, you know, anywhere from $25 to up to $4,000 is a big difference. There are also monoclonal antibodies approved for the treatment of Ebola, COVID-19, and also HIV. So with regards to the current pipeline of preventative monoclonal antibodies in active clinical trials, we reviewed clinical trial registries. And just to say that active was defined as any trial that was registered after January 2015. So it doesn't include products that are no longer in development. And we have not included monoclonal antibodies for treatment here, although, you know, there are obviously some of those as well. So we identified one candidate in clinical development for chikungunya, and this is actually a mRNA that codes for the monoclonal produced by Moderna. We also identified 3 Ebola candidates for pre-exposure or post-exposure prophylaxis to Ebola. There were 10 candidates for the prevention of HIV that we found in our search, 4 malaria monoclonal antibody candidates, another rabies candidate in phase 3 clinical trials. And for RSV, there are 3 monoclonal antibodies that are actually long-acting in nature, varying from phase 1 to phase 3 clinical development. There's also a Staph aureus candidate in phase 3, and then there's a couple more SARS-CoV-2 candidates, a yellow fever monoclonal antibody, and a Zika monoclonal antibody product in development. So it's a fairly rich pipeline, actually. And obviously, you know, I mentioned before that these products are expensive to produce, but we really do see that they do have a niche application for the prevention of some infectious diseases. One of the main advantages of monoclonal antibodies is that they offer quick protection, whereas compared to vaccines, the immune response is obviously not immediate, and sometimes it takes more than 1 dose to mount a full immune response. When compared to drugs for prophylaxis, they may have fewer side effects, which is also another advantage. And we really see the potential application in the prevention of infectious diseases during periods of high risk, where vaccine development has traditionally been quite challenging. And I think a really nice example of this is the Rsv monoclonal antibodies, because vaccine development has been so challenging, especially for very young immune-naive infants. And having these long-acting monoclonal antibodies, which basically is given at birth or soon after birth or just prior to the onset of the RSV season, really just offers several months of protection to infants when they are so vulnerable, and just really delays that first RSV infection until they're a little bit bigger, their lungs are more developed, they're more robust. We also believe that they do have an application to help address antimicrobial resistance. And obviously they have applications for use in epidemics or pandemics, and that's been demonstrated through the COVID-19 pandemic. They also present an alternative to the blood-derived immunoglobulins used for rabies post-exposure prophylaxis and for diphtheria, because they offer more standardized production than the equine-produced products in particular. But as I mentioned, they do have disadvantages, and the cost is probably the biggest of those disadvantages. They are just more expensive to produce than vaccines and chemically synthesized drugs. The protection is also short-lived, although with the extended half-life products that are now in development, they do offer several months of protection, which is an improvement. Compared with drugs, they obviously have to be administered by healthcare workers because they're either injected intramuscularly or subcutaneously. or they're administered by intravenous infusion, which is much more complicated. And of course, the clinical development for some of these products, especially for those that are rare or have sporadic epidemiology, is pretty challenging. But I think that's true for all of those pathogens, be it drugs or vaccines that you're trying to develop. And so I mentioned that the immunisation department at WHO has an interest in these products. And so what we've started to do is develop preferred product characteristics. And these preferred product characteristic documents really aim to encourage innovation and to promote the development of products with a view for using them in low and middle income country settings. So they basically describe what the preferential attributes should be, including indication, schedule, target population, route of administration, programmatic suitability, as well as covering some of the considerations about the data that needs to be collected in terms of efficacy and safety. So far, we've developed preferred product characteristics for monoclonal antibodies for HIV prevention. And that document was just published a couple of months ago, and there's a link to that just shown there. And last year, we published preferred product characteristics for monoclonal antibodies to provide passive immunisation against RSV in infants. And WHO currently has a preferred product characteristics document under development for prevention of malaria in infants and young children. So in a nutshell, that's sort of our view of how these monoclonal antibodies might be used for the prevention of infectious diseases. I'm going to hand it over now to my colleague, Richard Ipsbrucker. He is from the Norms and Standards for Biologicals unit here at WHO, and he's been working on some of the more normative guidance documents around infectious disease monoclonals. So with that, I'll hand it over to you, Richard.
Thank you, Erin. One moment while I share my screen. And hopefully everybody can see that, and that is in proper presentation mode.
Looks good. Thanks, Richard.
Great. Thank you, Erin. So as Erin mentioned, I'm in the Norms and Standards for Biologics unit. And for those who are not familiar with this, this is the group at WHO which is responsible for establishing the guidance documents and biological reference standards that are related to the quality, safety, and efficacy of biological therapeutics, such as monoclonal antibodies, as well as for vaccines. And these guidance documents that we have several purposes, but their primary role is really to provide the key principles and specifications on which regulators can set their national requirements and guidance, and these are also important for use for the WHO pre-qualification program. And these documents really facilitate the scientific and risk-based evaluations, as well as the post-approval change management for these types of products. Because WHO is an international organization and that these guidelines really are available to everyone and they're open, these help to increase the international harmonization on product specifications. And they also have served, as we found, as they really improve the communications and expectations between industry and regulators. And so they really sort of serve as that, they can serve really as a mediator as industries approach the regulatory authorities in their countries. And these guidelines are really meant to complement the existing national and international regulations and guidelines, and also to provide guidance where none may exist, such as in those countries that just may not have that regulatory capacity. All of our guidelines that we post at NSB are written by a group of international experts that are invited to join the drafting group. And all of our guidelines are also posted through a minimum of 2 public consultation processes. And so we managed to get an input from, or the available input from around the world. And then prior to their adoption, they are reviewed by the Expert Committee on Biological Standardization, or ECBS, and once they've adopted them, they're published as annexes within the committee report, which is published as a Technical Report Series, or TRS. So in regards to monoclonal antibodies for infectious diseases, we have quite a large number of guidelines that are relevant to them. And the bulk of these are really related to their quality control. And this ranges from like recommendations for the evaluation of animal cell cultures as substrates, all the way to the good manufacturing practices, GMP for biological products, a good laboratory, and good clinical practices. But perhaps one of the more important ones, or the more relevant ones that we currently have, is this one, which is the Guideline on Quality, Safety, and Efficacy of Biotherapeutic Protein Products prepared by recombinant DNA technology. So that guideline was adopted in 2013 and is published in the subsequent years, so in 2014. And it's a really good but general guideline. It provides a strong regulatory foundation for the control and evaluation of biotherapeutic proteins, including monoclonals and antibody mimetics. And I'll sort of come back and touch on this again a bit in a few slides. But because this is such a general guideline, and it's also pretty much all biotherapeutic products, and regardless of the indication, for many regulators who are perhaps less experienced or who have less exposure to these types of products, it can be difficult for them to extract the monoclonal antibody specific information or regulatory guidance from it. And it also has very limited details and specifications which are unique to the monoclonals. And one problem that we found also within the past few years is that it is written really in the context for use, or of the use of monoclonals for non-communicable diseases, sort of cancer and immunology. And so applying it to the conditions of infectious diseases is again a little bit more tricky and really requires a bit of understanding of regulatory nuance. And this is really particular for things like the non-clinical development and clinical trial considerations for pre or post-exposure prophylaxis and therapeutic indications. So coming up to the beginning of the pandemic a few years ago, we realized that we were having some of the regulators were approaching us and realized that there was a bit of a problem because at the time, so in the summer of 2020, there was already well over 100 monoclonal antibodies to COVID-19 that were in development. And also at that point in time, there had been a number of monoclonal antibodies, and so Aaron had mentioned this earlier, had sort of highlighted some of them earlier, that were already either in clinical trial or already licensed to infectious diseases such as RSV or rabies. And so we approached the expert committee at their meeting in October of 2020 with a proposal to generate a guideline on the quality, safety, and efficacy of monoclonals that were intended for infectious diseases, and then to draft separate supplements to that guideline, which would provide any additional disease-specific information. And they approved, or they agreed with this proposal. And so we conducted a review then of our range of the WHO guidelines, which we had and which I just showed you earlier. And based on the outcomes of that review and some internal discussions that we had, we revised the proposal slightly to ECBS at their December 2020 meeting, and we split that project into two separate guidelines. And so the first is the guideline on the production and quality of, the production and quality control, essentially, of monoclonal antibodies for use in humans, and this is regardless of their therapeutic intent or biosimilar. And this was, and this means that you can use, this is a guideline that is really applicable to all monoclonal antibodies, because we realize through the Wellcome IAVI report on access to monoclonal antibody therapies, which was published that same year in 2020, that summer, that there had been a lot of disharmonization in the expectations and regulatory guidance surrounding the expectations around monoclonal antibodies. And so we felt that this first guideline and establishing one that was really applicable to all of them would be very helpful. And the second part of this project was then to develop a guideline on the safety and efficacy of monoclonals that's specific for the prevention and treatment of infectious diseases. And to that one, we would then later draft disease-specific supplements. So earlier this year, the first of those two guidelines was adopted by the expert committee on biological standardization, so ECBS, and this is the guidelines for the production and quality control of monoclonal antibodies and related products intended for medicinal use. This was published in this year's Technical Report Series 1043, and the link to that TRS is at the bottom of this page. And so the scope of this document includes monoclonal antibody products, or those products that are based on an immunoglobulin scaffold. So that's whole monoclonal antibodies, co-formulated or cocktails, whether they're conjugated, bispecific, biosimilars, mAb fragments, and so on and so forth. It applies really to a range of expression systems, whether it be in vivo, or in transgenic plants. And it also is applicable to pretty much all manufacturing approaches. So traditional manufacturing, it discusses platform technology, continuous manufacturing, or single-use manufacturing conditions. And it is also applicable to all routes of administration. In the drafting of this, we realized we had to add some exclusions or limit some of the scope of that. And the first was obviously the nucleic acid-based platforms, as the quality control and manufacturing principles around nucleic acid-derived monoclonals is extremely different, and it really doesn't apply for this. So that was out. We also felt, or the drafting group also felt, it was important that the antibody mimetic proteins, so things like affimers and DARPins and so on and so forth, really didn't quite fit, although much of the general principles sort of apply here. They really felt that the prior documents, so this guideline, was a little bit more appropriate for those antibody mimetic proteins. So the guideline itself is actually quite basic. Here's just a section of the table of contents. We'd have some terminology, and we have some general regulatory considerations in there. And throughout the document, we do discuss some parts which we normally would not. So we do mention parts that may be important for the manufacturers to consider during their development process, and that would be important for later in terms of their, to consider in terms of their marketing authorization applications. And then the bulk of the guideline is on the manufacturing recommendations, and this goes everything from reference preparations and the control of source materials all the way down to stability testing and storage. The principles really follow this, which is a very basic outline of common manufacturing practices, and it sort of follows this flow from the initial seed, whether it be a plant seed or a cell culture or even in animal, so in vivo production, right down to the final fill. And I won't go through all of these details in here. It also applies to a wide range of expression systems, as I mentioned, and it's really important to note, and the document notes that the target product profile of monoclonal antibody will really detect the expression system to be used. So those two really are sort of linked. And it's important to realize that some monoclonals really require a mammalian expression system in order to produce a biologically functional product. And this is particularly so for the glycosylation aspects of that, whereas other mAb products with simplified glycosylation, or those that are lacking it, can be produced or suitable for lower organism expression platforms. And so, as I mentioned, we do allow, and we do provide room within this guidance for the in vivo expression, although that is, such as the originals, which was done in mouse ascites, although we do discourage this mode of production. The bulk within the commercial space, the bulk of production that is currently within the commercial space, is really from NS0 and CHO cells, and so the mammalian cell lines. Although we can see that coming down the pipeline, there are other expression systems, and I know that today some people will be discussing some of these. What was really interesting within the discussion with the expert committee when we presented this was a bit of a word of caution for the manufacturers who are looking to produce monoclonals in plants, particularly those who are producing them in tobacco plants. These are by far the most advanced plant species, or the best known in terms of their development for production of monoclonal antibodies. But it was noted that many of the manufacturers that use these or that are producing in tobacco plants have funding and are associated with the tobacco industries and companies, and if that is so, that their products would not be available through the WHO pre-qualification program. We've already seen that an issue in one of the vaccines for Ebola, or not for Ebola, pardon me, so for COVID. That became an issue with the WHO pre-qualification program. So that's just sort of a little bit of a caution to anyone out there who is developing it within tobacco species, just to be aware that if there is clear funding or resources provided by the tobacco industry, that this may cause an issue. Although if those are just being produced within a country or being sold directly to the country, then that pretty much, that point is moot. The second guideline that I mentioned is that for the non-clinical and clinical monoclonal antibodies, and I'm happy to see that that there are several participants today that are on the drafting group for this document. So the first version of this, or the first draft of this document, which is the guideline, currently called the Guideline on the Non-Clinical and Clinical Evaluation of Monoclonals and Related Biological Products Intended for the Prevention or Treatment of Human Infectious Diseases. This is currently live at this web link. It is open for public comment, so anyone here may provide their comments on the document. However, those comments must be received by me by close of business on the 28th of October. Here's a general list of the contents that we have on there. So it has a general regulatory considerations section in there, some terminology. It goes through the in vitro and in vivo non-clinical evaluation related to infectious diseases and clinical evaluation as well. So everything from some general considerations in phase 1 studies right through to phase 4 or post-marketing studies, as well as pharmacovigilance and risk management planning. For the scope of this does include all, like the prior manufacturing guideline, it does include all monoclonals which are based on immunoglobulin scaffolds, but it also does include the monoclonal or the antibody mimetic proteins. However, this drafting group also felt that it was important that that this guideline does not have within its scope the nucleic acid-based platforms, as those really come with other issues. And in terms of the disease-specific supplements, these have not been started yet, but these supplements will be intended to be short documents, so very brief ones, whereas the guidelines can tend to be 30 to 50 pages long. We're looking at something that might be 5 to 10 pages, which provide additional details that would go along with this guideline, with the non-clinical and clinical guideline, and that they might provide a little bit more, the finer details relevant to the development and assessment of monoclonals for a specific disease. And so, for example, this may have information on the infecting agent, its pathology or variants, it might be relevant, its epidemiology or transmission, if there are or are not any international reference materials, any information on antigens or epitopes that's known, such as those might be involved in neutralizing activity or enhancing. It should discuss whether there are any known in vitro models and assays or relevant animal models to the disease, as well as any human challenge models. And hopefully, if there's any information or data available on the clinical evaluation of monoclonal antibodies for the disease or even vaccines to the disease, this would be helpful in providing some additional supplementary information. And so at the moment, we have 5 supplements that are being planned. The first 2 will be for COVID-19 and RSV, and we hope to start drafting those closer to the end of this year or early in the next year. And we are looking for drafting group members for these 2. And then later next year, we will continue with the other 3, so for rabies, malaria, and HIV. And undoubtedly, as we get familiar with the drafting of these supplements, there no doubt will be additional supplements being drafted. And really the advantage of making these supplements and making them short is that it means they're much more flexible in being able to revise in the future. And hopefully they'll also be much less expensive for us to develop. So if there is any additional information needed, this is the link to our website for the norms and standards for biological therapeutics. And it provides the links to our guidelines, if you dig around in there, and all of the TRS publications, which can be found under the ECBS tab. And I will leave it at that. Thank you very much.
Thank you very much for that, Richard. Really important, and I would encourage anybody that's interested to do provide comments to that document using the link that Richard displayed on his slides by the 28th of October. I see there is one question in the Q&A, but it seems to have been addressed by Larry in the chat. So you're off the hook, Richard, for questions.
Great. Thank you very much. Lucky me.
Thank you. I'm going to hand it over now to Shelley Malhotra from IAVI. She's the Executive Director on Global Access at IAVI to talk about some of the work that they're doing. Over to you.
Thanks, Erin. Just pulling up my slides. Okay, perfect. So I wanted to focus on how some of the efforts we're working on at IAVI to work towards accessible monoclonal antibodies end-to-end along the product development continuum. And I wanted to start out first by thanking our donors and also by setting the stage a little bit. This slide highlights some of the leading causes of death in both low-income countries on the left and lower middle-income countries on the right. And anywhere you see a star, that indicates a disease area for which there is either an approved monoclonal antibody or an antibody in the pipeline. And this is really just to highlight that there are several diseases of important relevance to LMIC settings for which there are antibody solutions either available or on the horizon. However, global access is currently severely limited. And this graphic is from a report that IAVI developed with Wellcome Trust a few years back. And it really highlights that the vast majority of antibody use is in a few settings, in high-income settings in US, Canada, and Europe, despite comprising the minority of the global population. And antibody use in Africa is only about 1% of global sales, and less than 10% of antibodies that are approved in the EU or US are even available in Africa. And so lack of availability and affordability are key challenges. On the left, this highlights, also from a report, highlights that there are a large number of antibodies that have been registered in the EU or the US, but only a minority of these make their way to Africa or other resource-limited settings. And price is a key factor, as has been mentioned. This illustrates on the right pricing for the US market. However, it's just an indicator of how exorbitantly priced these antibodies can be. And so price and lack of availability are key challenges. So I just wanted to clearly paint that picture before talking about some of the efforts at IAVI to work on expanding access to monoclonal antibodies. So our overall mission is translating scientific discoveries into affordable, globally accessible public health solutions. And on the antibody front, we're working to do this by focusing on developing products that will be acceptable, affordable, available, and widely adopted. And some of the key barriers to access that have been mentioned already and that we're focused on are product profiles, the business models and pricing approaches, the production costs, which is the focus of this meeting, and regulatory strategies, as well as some of the policy financing and implementation gaps. And so I wanted to talk about some of our efforts in each of these areas. So as Erin mentioned, currently, a lot of the monoclonal antibodies on the market are requiring IV infusion based on their volume requirements, and they are expensive to deliver and require significant infrastructure, including skilled providers. Their dosing schedules often require frequent clinic visits and often, and most part, carry cold chain requirements. And so what we're really focused on is working to optimize the profile of antibodies early in discovery, so that we're working towards highly potent antibodies that can enable low doses to be delivered through injection. And this can enable task shifting and use in a broader range of settings. We're also looking at half-life extensions for less frequent administration and strategies for moving towards more thermal stable formulations using lyophilization technology. And in addition, we're really working to understand what the end user and stakeholder perspectives are on product preferences for future antibody products. Erin mentioned the preferred product characteristics work for monoclonal antibodies for HIV prevention. That was a joint initiative between WHO and IAVI with support from Wellcome and USAID. In addition, we're engaged in assessing the feasibility, acceptability, and optimal delivery strategies for future antibody-based products. And on the bottom left, I've listed a few of the projects we have ongoing to look at acceptability and feasibility considerations, one being the Uptake Initiative supported through EDCTP, and then the other, the USAID-funded advanced project. We have a series of studies looking at feasibility and acceptability factors. And this is really meant to steer our product, optimization, design, delivery, and development approaches. I also noted the challenges around the current business model for antibodies. The current market, antibody market is based on kind of a low volume, high margin model. And the pricing strategies are not developed with broad access and affordability in mind. The entry of biosimilars has, exerted significant downward pressure on pricing. And we can see this with the introduction of Indian biosimilars, which are on average discounted by 57%. And you can kind of see this trajectory on the left in downward pricing with the introduction of multiple biosimilars. But you can see also that this happens after a significant delay following a long period of marketing exclusivity. And there are regulatory and technical challenges, as most folks on this call know, in terms of showing analytical and functional similarity between innovator products and biosimilars, which can delay access because unlike generics, they're not identical copies and can be much more expensive to develop. And so we see, lengthy delays in terms of reaching these affordable antibody-based products. And so this is a topic that we would like to delve deeper into. What are some novel partnership strategies that can kind of address these unique drivers of antibody costs and advance an alternative business model paradigm for antibody access? And so in collaboration with Unitaid, Wellcome, and Medicines Patent Pool, we will be co-convening a workshop on novel business models for sustainable, scalable, and affordable global monoclonal antibodies access. And this will be taking place in the first quarter of next year. And objectives are really to explore innovative partnership approaches and strategies and to define a set of priority interventions and enablers for novel business models. Regulatory barriers are also, or delays are also a key challenge. And I think, Richard spoke to some of the capacity strengthening and guidance efforts that are in place to help support regulatory pathways for monoclonal antibodies. But we also see delays in actual filing between high income settings and resource limited settings. And so some of the strategies, we're prioritizing in our efforts to minimize these delays are ensuring that the terms in our licensing agreements prioritize broad and timely filing in LMIC settings, that it's not an afterthought, but that it's core. And that we have early engagement with both stringent, in quotes, reference regulators, as well as endemic country regulators and networks to ensure the study design and clinical development plans are conducive to individual licensure. In priority settings, we are engaged in some capacity strengthening efforts through our advanced project and through our plans to support, to engage in some of these joint review pathways And then advocacy is also really key in order to prioritize timely access in LMICs. This is just a brief snapshot of some of the recent advocacy efforts that we've been engaged in, issuing reports on topics of antibody access and articles, as well as several webinars and panels, really trying to mobilize attention around this issue of access. And also importantly, it's going to be that it's also important that monoclonal antibodies are integrated into policy, financing, and procurement mechanisms. And this has not yet been the norm. So on the left, you can see that for the most part, when it comes to lower middle income countries, antibodies are not yet integrated into national financing systems. They're also not broadly integrated into any of the global pooled procurement or financing platforms. I think a key exception is the work through Act A to extend access to antibodies for COVID-19 therapy. And this is a great first step, I think, in normalizing the fact that we should be integrating antibody-based therapies and prevention products into some of these global mechanisms. And some key enablers to that we've been working on, of course, are building awareness, helping to define what the pathways and strategies are for adoption of antibody-based products, both at the global level as well as national level, and then also really prioritizing gathering the clinical and programmatic evidence that's going to be needed for adoption concurrently. So evidence on cost effectiveness, programmatic suitability, et cetera, so that evidence is there to inform adoption decision making. And I won't touch on this one too much because this is the focus of the meeting, but just to say that, we're engaged in a broad range of efforts along the product development continuum, but one of the most important efforts is going to be the work of this group here today in really exploring how we can bring down the cost of antibody manufacturing, because at its core, it is more expensive than other manufacturing processes, and so we need novel strategies for how we can bring down this cost of goods. And so just to summarize, efforts are really needed along the product development continuum, working early on in terms of ensuring highly potent, optimized products with longer half-life, optimizing our manufacturing to processes to reduce cost of goods, ensuring we have formulations that are conducive to broad use, and working towards low-cost regional manufacturing strategies. And then finally, defining a pathway to access by stakeholder-informed target product profiles, ensuring we have acceptable products, affordable and cost-effective, and then making sure that we are prioritizing filing in LMICs and integrating into the policy, financing, and procurement pathways that are going to be so important for access, as well as raising awareness around this issue of monoclonal antibodies access. And that is it for me. Thank you very much.
Thank you so much, Shelley. No questions for you in the Q&A. There have been a number of questions raised about WHO pre-qualification of monoclonal antibodies, which I see some of my colleagues have replied to. But just for those of you who are not watching the chat, there are several monoclonal antibodies now approved by WHO. There's the biosimilars for trastuzumab for breast cancer and rituximab. And then there is also one monoclonal antibody for COVID that has been pre-qualified. So just checking the Q&A, no questions for you. Thank you, Shelley. All right, well, I'm going to hand it over now to Vasil Kaiser from the University of Sydney to give a bit of an overview of the current production technologies and the pipeline technologies that are under development and what the pros and cons are of each of those. Over to you, Vasil.
Thanks, Erin. Could you please confirm that you can see my presentation?
We can, but
full
it's
screen?
yes, we can see it in presentation mode. Thank you.
Okay, not the presenter mode. Okay. I'll switch off my video.
Oh, sorry, it was in presentation mode, but now it's not anymore.
Okay.
Yep, all good. Thank you.
Okay. All right. Hello, everyone, and thanks for joining this timely meeting. And I'd like to thank WHO, particularly Erin, for providing this opportunity. In this talk, we will be looking at the production technologies of monoclonal antibodies or MABs broadly and briefly stability issues. My talk will cover a wide range of topics without going into details, and it will only scratch the surface. We will go through the following topics briefly. First, we will compare small molecules and antibodies and point out some of the critical differences between them. Manufacturing of MABs will be summarized. Basic structural features will be mentioned. Other important considerations when developing MABs will be covered, such as hybridoma and phage display technologies, protein expressions, post-translational modifications, etc. I'll briefly mention about glycosylation as well. And protein aggregation is arguably the most detrimental degradation for protein-based biotherapeutics, and including antibodies. So we will look into aggregation issues and stable formulation, how we can achieve stable product through these as well. In this table, we summarized main characteristic differences between antibodies and small molecules. For example, antibodies are very large, complex proteins with about 150 kilodalton molecular weights compared to small molecules that are typically less than 500 daltons. This is a busy slide, so I will not read them, read line by line. Antibodies need to be produced using mammalian cell lines mostly, especially if it is a full-size antibody with many different processing steps, but small molecules are usually chemically synthesized with fewer processing steps. Characterization is often difficult for antibodies, and they are not very stable at high temperatures, whereas small molecules can be well characterized and they stay stable for a long time at room temperatures, at least most of them. Antibodies are usually formulated as injectables. This is also true for other biologics as well, but small molecules can be prepared as solid dosage forms. Of course, there are many advantages of antibodies compared to small molecules, such as long serum half-life that could be several weeks, sometimes months, and they are highly specific for their targets, and therefore we see fewer side effects compared to small molecules, but of course, those side effects could be immunogenic as well. Disadvantages of antibodies include, as mentioned before by previous speakers, their high costs and heterogeneity as well, partly due to post-translational modifications. Here are top-selling drugs with annual revenue in billions of dollars. Even though only a fraction of prescribed medicines are biologics, they are the top-selling medicines. This is especially true for monoclonal antibodies. They have gradually become the top-selling drugs within a short period of time. As you can see here, there are over 100 antibodies on the market for different indications, but thousands of antibodies and antibody-based products are in clinical trial altogether. As you can see, the growth is exponential. This trend very likely will continue over the next few years. The history of antibodies as pharmaceuticals is shown here. Serum products and antitoxins have been in use for a long time already, since 19th century. And these products contain polyclonal antibodies. Some of them are still in use. Monoclonal antibody therapy pretty much started mainly with the development of hybridoma technique in the mid-70s. 10 years later, in mid-80s, humanization of antibody was achieved with the advancement of phage display and CDR grafting technologies. First monoclonal antibody was approved in 1986 for use in preventing kidney transplant rejection. And first humanized antibody, trastuzumab, was approved in the late 90s to treat HER2-positive cancer, mainly breast cancer, but nowadays some other types of cancer too. As mentioned, the antibody market today is huge, probably the largest of any other drug class. This is showing the structure of an antibody, IgG1 antibody. The structure can be divided into two fragments: antigen binding FAB region on top and FC fragment. FC fragment contains glycosylation, which is important for structural stability as well as for other important characteristics. In addition to full-size monoclonal antibodies, there are other formats as well. Some of the most important ones are summarized here. These formats are not used for infectious diseases at the moment, but we expect that some of them will be adapted in the future. Antibody drug conjugates, or ADCs, as one can see here, for example, is particularly for cancer indications. There are other types such as bispecifics, again, mostly cancer, but recently for several other indications, several bispecifics also have been approved, and various other types of formats such as fusion proteins or fragments. So, how do antibodies work? Some of these points were already mentioned before. First of all, they are large proteins. Most of them are IgG1 antibodies, and they have around more than 1,300 or more amino acids. Due to the reasons we talked earlier, they are usually formulated as injectables and administered intravenously or subcutaneously. And they are highly specific for a particular target, so this makes them ideal choice for preparing targeted therapies. They bind to their targets through variable domains from FAB domain, FAB fragment, but rest of the antibody structure is conserved. So therefore, therapeutic antibodies share a great deal of common features. Their targets could be anything, for example, surface receptor or a protein in a cancer cell or a virus. They do have very long serum half-lives because they bypass renal filtration, mainly due to their large size, and also some are recycled. Antibodies don't present side effects that we see with small molecules. However, especially non-human antibodies could be quite immunogenic, and other types of side effects, some serious, can be observed with antibody therapies. In fact, all these characteristics we mentioned so far also depend on the origin of antibodies, whether they are human or humanized or chimeric. Different formats are shown here again for our discussion for infectious diseases. Naked or full-size antibodies at the moment are important, but other formats might be important in the near future. Let's have a look at the manufacturing steps. This is quite complex. Now I'll briefly talk about some of these approaches. I'm extremely simplifying here and in the later slides as well. So if we briefly divide into two different steps, the first one is developing a candidate. Sometimes this is a library for one type of target, and one has to choose one of these candidates to develop further. During this time, we have to be considered considering several different important aspects, such as the high stability, what type of formulation we need to have, buffer type, salt, and so on. And all of these tend to be expensive and time consuming because most of the steps are relying on trial and error approaches. And some of these considerations and challenges are listed here. I hope you can see it. Again, we will not go into details, but for example, disease indication, what type of modes of engagement, and what type of formats are important, post-translational modifications, whether it is stable or not. All these issues are actually all the way from discovery until field finish challenges can be observed. But new developments also exist to address these manufacturing challenges. Again, you can have a look at these publications to read the details, but new advancements can address some of the challenges that we observe, and these are very well known challenges in industry as well as in research labs. Approaches for the development of antibodies are shown here. What is important to mention is that these methods are applicable for the development of both therapeutic and pre-exposure prophylaxis antibodies. And I'll go through them one by one without, again, going into details. So a traditional mouse hybridoma technique starts by immunizing mouse with desired antigens that triggers an immune response, and it generates a lot of different B cells. And these cells are harvested and fused with a myeloma cell to produce hybridoma cells. And these cells persistently secrete antibodies, of course. You need to screen which type of B cells, hybridoma cells you are after, and select the lead, and then generate chimeric or humanized antibodies. There are advantages of disadvantages, obviously. All those methods that I will mention. This is a well-established method. As I said, it was developed in mid-70s. It is relatively low cost. It has high specificity, but of course, disadvantages include it takes a long time, and immunization is needed. You can also use chimeric as chimeric antibody, but nowadays most of the antibodies require some sort of immunization studies. And what it means is, if a mouse antibody is generated, CDR grafting technique can be used to generate chimeric antibodies. And if you mutate it further to make it more human look, human sequences, you can actually humanize the antibody. The second one is phage display antibody. A library is used to select antigens of interest. After several rounds of biopanning, immune positive phage clones are screened by ELISA method, and then DNA sequences are analyzed to construct and express human antibodies. Obviously, the main advantage of this is this is a human antibody now. It's quite cost effective. It is relatively easy to use. It can be high throughput, and you can generate antibodies for a wide range of targets and maps from a wide range of B cells. And there are some disadvantages as well. The main two probably are the PTMs, post-translational modifications, are limited. Also, the full-size monoclonal antibody expression requires other assays. Relatively new methods include transgenic mouse, similar to mouse hybridoma technique or single B cell method. This time you need to generate a transgenic mouse that will generate full human antibodies. This is a good method, obviously, because it will generate full-size human antibodies and high-quality antibodies with appropriate post-translational modifications. But obviously, there's initial investment cost as well as transgenic animal is needed, and you have to have good animal facilities to be able to generate this. The last one is single B-cell techniques. Some of the COVID antibodies actually use this method to generate monoclonal antibodies. From infected or vaccinated donors, you can get samples, and then you can generate B cells by flow cytometry. And the advantages obviously is fully human antibodies, good specificity, affinity, and diversity, but the main disadvantage is you need an infected and recovered volunteer for this method. And this is pretty much summarizing all these methods. So I thought this is from this publication, this figure. So I thought I should cut it down to digestible sections. Without going into too much detail, let's have a look at the overall manufacturing steps. Again, this is a very simplified diagram. So only some of the processing steps, mainly critical ones, are shown. So you obtained your DNA from one of the previous methods, let's say, and then you can use mammalian cell lines mostly to generate these antibodies. Now, there are some differences between transient transfection as well as stable cells, stable cell line that is used in industry. However, similar steps exist, so I will not distinguish in this talk between those two systems. Using, as I said, using mammalian cell line, whether it is CHO, Chinese hamster ovary cell line, or other cell lines, you can generate a lot of antibodies. At the moment, industry is dominated by CHO cell lines. Once you secrete a lot of proteins, then you separate it by purification, whether protein AG purifications, but the most elegant one probably would be antigen purification. And generally speaking, additional purifications are also required, such as ion exchange purification, sometimes size exclusion, chromatography, and this purified antibody now can be formulated with different formulation conditions, and the successful formulation is used as drug. Of course, Richard mentioned viral this method as well, these steps, and I have skipped most of the other methods that are generally used, such as viral removal and so on. More complex proteins, such as antibodies, would be under scrutiny compared to smaller molecules or peptides. However, more complex molecule means, generally speaking, less conclusive data, so characterization might take longer, or more experiments might be required. And these characterization, antibodies need to be characterized by various different methods for different aspects, including chemical physical stability that we will look in a minute. Amino acid sequence, of course, post-translational modifications, and so on. And out of different types of post-translational modifications, glycosylation probably is the most important PTM, as it cannot be controlled easily, and it provides heterogeneity, and it does affect various aspects of the protein, including stability and solubility. So it has to be characterized quite well. And as I mentioned, glycosylation is located in the FC domain, and it can be quite complicated depending on the protein. The Other important aspect is the protein aggregation. Probably protein aggregation is the most common degradation method for protein-based therapeutics. It is a function of many different things, such as initial protein concentration, initial aggregation concentration, storage conditions, temperature, and so on. So it is not easy thing to control. But it has to be less than about 5% to be able to be marketed for any protein drug, especially monoclonal antibodies. It is important to characterize and try to control because aggregates could be immunogenic. And generally speaking, although native aggregates, native proteins can form native aggregates, most of the time the issue is the partially unfolded protein aggregates irreversibly, and that is the issue. When it does that, the outcome is non-homogeneous behavior. In other words, you cannot predict what's going to happen at low temperature using accelerated studies from high temperature data. There are different methods that one can use for this. One of them is a VFD function, for example. But generally speaking, this is one of the issues in protein-based, actually all biologics-based products, that we cannot really extrapolate easily what we are finding at high temperatures to low temperatures. A stable formulation is also a must for all medicines. So this step is crucial for monoclonal antibodies because long-term stability of the product depends on it. Proteins are marginally stable at a relatively narrow pH. So keeping the solution at pH, keeping the solution pH constant, we use different buffers such as histidine trees. Keeping pH constant is also a safety issue for injectables. Again, the slides will be distributed, of course, so I will not go through this, but formulation is one of the difficult aspects of monoclonal antibodies, obviously, as it will affect the stability of the product. And stability is relates to various physical, chemical, and microbiological reactions that may change the original properties of the preparation during storage, transport, or use. For biologics, including antibodies, physical stability could be denaturation, for example, of the protein, or aggregation. Chemical stability could be hydrolysis or fragmentation of the protein, oxidation, deamidation. Basically anything, any reaction that forms new chemical bonds or breaks existing bonds would be categorized as chemical degradation. And the characterization and stability studies, of course, are a requirement for approval by regulatory agencies as well. How can we really make them stable? So I will focus on mostly for aggregation here in the next couple of slides. So we can ask whether we can change amino acids if it is especially early phase, in the early phase development. If the answer is yes, then we can mutate surface amino acids. We can add any additional glycosylations. But if we cannot change, we can maybe change formulation of the product. I will show you some examples. Here, in order to achieve this, we used computational method a number of years ago. And by making point mutations, you can see that some of the variants behave better than the wild type. In other words, they aggregate less. So by changing one or two amino acids, you can achieve a better product that is less aggregation prone. This is all, of course, a good achievement because we are talking about more than 1,300 amino acids. Here, you cannot really do this easily by changing one by one. Additionally, what you can do is you can add glycosylations, additional glycosylation. When we do that, similarly, this is a similar monomer loss. several variants were more stable, they aggregated less. And this can be seen easily from different melting temperatures of these variants because antibodies have more than one type of melting temperature. If you cannot do this, you can do formulation development by adding different additives into it, whether it's a mixture of several different amino acids or new type of additives. We found out that, as well as others, new things such as ionic liquids, for example, could provide very good stability for antibodies. Maybe these kind of things can be considered for developing monoclonal antibodies for infectious diseases. Take-home messages are we need new approaches to reduce the overall cost of antibodies and to reduce the disparities of affordability between high and low income countries. And these new approaches might be essential for to be able to achieve this. And new methods could be new protein expression systems or systems that yield better protein product, or existing systems can be changed to address some of the issues such as glycosylation. Subcutaneous delivery seems to be the preferred method currently, but this requires high protein concentrations, which lead to protein aggregation and sometimes high viscosity. So new formulation strategies are needed. And lastly, we should move or try to move beyond trial and error approaches, of course, and start doing rational development. And this is only possible if there is a front-end investment. With this, I'd like to thank you for your time. I also would like to thank my current and previous postdoc students, collaborators, and the funding agencies. If it is not too much to ask, I'm also looking for a postdoc to work in this field. So please do share with potential candidates.
Thank you very much, Faisal. So if anybody wants to go and do a postdoc, you've got the link there to apply. There has been a few questions posted in the Q&A. So the first question is, are there places in the traditional CHO cell-based manufacturing that could be improved or optimized and modified to reduce the costs? And I think you did allude to some of the things around aggregation and that kind of thing. But did you want to provide a little bit more thoughts about sort of the future of CHO cell manufacturing?
Yeah, I was. I realized I was over time, so I actually passed through quite quickly those aspects. So at the moment, with stable cell line in industry, more than 10 grams per liter can be achieved. That is a very good yield over the years. I think they optimized it to get a really, really good yield. So maybe cell line optimization can continue, but up to certain level, I suppose. So for every product, one has to do this kind of optimization anyway. But what I didn't have time to mention is maybe as a new product, if we don't need a full-size monoclonal antibody, maybe we can consider using other types of cell lines, even microbial cell lines or yeast, or even plant to generate these antibodies to reduce the cost substantially. But in that case, we have to be mindful about the aggregation issues as well as other stability issues, so that formulation has to be done a little bit better than what we are doing at the moment. So yes, there is room for improving that.
Thank you. And there's also a question about half-life extension and whether or not that's a key consideration.
Yeah, this is an excellent question as well. And it is If it is not fully human antibody, then half-life will be much reduced. But also, if it is fragment, such as FAB only, the half-life will be reduced. So yes, absolutely, this half-life extension also is a key consideration, especially for those antibodies that are used as neutralizing antibodies. What you want is you want to circulate a lot of antibody for a long time so that you don't have to keep injecting it as well. That will be detrimental for cost, as you mentioned as well in your talk, Erin.
Yeah, and I think we're gonna hear from about RSV monoclonals and long-acting products that are in development later on. So yeah, I think that's going to be a real game changer for some of these products. There is also a question about bispecific antibodies and how that might reduce the costs of monoclonals.
Yeah, so antibodies work in different ways. Sometimes they can just bind to a pathogen, in this case, for a virus, for example, and then virus is prevented entering into cells. But also they can engage with immune cells. So by generating a bispecific that binds to two different cells simultaneously, we might be able to engage immune cells to destroy pathogens relatively easier. This has to be shown. We already know this for cancer, for example. It can easily be done. Well, not easily be done. There aren't many products out there, but a proof of concept is there because there are a couple of antibodies in bispecifics in clinical usage. But for no one has, to my knowledge, no one has published and showed that bispecifics, the bispecific formats can be used for infectious diseases. But I'd like to say that this has a lot of potential. So we need to keep working on these kind of things. I mean, I don't know what kind of so maybe immunology, those people who are working with immune system could answer this question a little bit better, but I do think that there is a great potential there, even though I cannot give more details at the moment.
Yeah, and I especially think for where, for viruses like HIV, where there is such great strain diversity, having sort of bi or tri-specific antibodies makes a lot of sense. There is a final question for you, and that's around whether or not you know if there are already any monoclonal antibody formulations that can be stored at room temperature that are approved.
Yeah, so if you lyophilize or freeze dry the product, if it is dry form, then you can keep it at room temperature for a while. And there are many biologics actually exist, even vaccines exist in this sense. So just prior to injection, you have to reconstitute this. But industry as well as researchers, and I believe even probably regulatory agencies, we would like to move away from lyophilization because this is also bringing additional cost. Because you have to do additional processing steps and so on. So yes, there are products exist, but even those products, lyophilized products, tend to be frozen or stored in, using or transported most of the time using cold chain process because you don't want to leave it for a long time at higher temperatures. But yes, lyophilized products exist already.
Thank you. There has actually just been another question that popped up into the chat. What about oral monoclonal antibodies?
Yeah, this is a dream of everyone. I think if we can develop products, antibody products, even any biologics actually that can be stored at room temperature as well as that can be taken orally. So this is a $1 million question. Our stomach as well as GI tract generally is not really a very friendly environment for these proteins, they tend to be degraded relatively easily. But there are some groups, we are working on this. There's a group in Harvard, as far as I know, working on insulin oral drug delivery using ionic liquids, using those additives, new type of additives that I mentioned. But there is no product at the moment. These are all in infancy stages. They will, we won't see anything in near future on the market.
Thank you. Well, we've gone a little bit over time, but not too bad. We're just 5 minutes behind schedule. On the agenda, there's a 10-minute break now. I'm going to shorten that to 5 minutes just to allow everybody to go and grab a cup of coffee. So we will restart at 3:25. So in 5 minutes from now, for those of you not on European Central Time. So thank you. We'll reconvene in 5 minutes. All right, so I'm going to hand it over now to Rip Ballou from IAVI to talk about HIV monoclonals. Rip, are you with us?
I'm with you. I'm just getting ready to share my screen. Bob, that was a fantastic presentation. Many of you know that I spent most of my career working on malaria, and it's fantastic to see this work going on. I'm going to talk about what IAVI is doing in the area of HIV. And in here, of course, we don't have a pathogen with a highly conserved target. We have a highly variable virus, and I want to thank all of our donors for the work that we do. But clearly there remains, as in malaria, a huge unmet need. The pandemic continues to cause extensive morbidity and mortality globally. Despite progress in reaching a majority of people living with HIV with antiretroviral therapy, there are gaps, and we have 1.5 million new infections and 650,000 related deaths in 2021. A particular subgroup of adolescent girls and young women living in low and middle income countries, especially in sub-Saharan Africa, are among the most disproportionately affected. And there are still 160,000 new HIV infections in young children, the vast majority occurring during infancy. And these are, I think, populations that, despite the presence of antiretroviral therapy, are still unserved and make a great case for an alternative approach involving monoclonal antibodies. The reasons to believe are the NIH and the HVTN recently completed a trial that's called the AMP trial of a first generation HIV BNAb called VRC01. And while that trial failed its primary endpoint, most researchers in the field believe that it demonstrated that that in the presence of an antibody that neutralizes a certain proportion of susceptible viruses, this trial demonstrated that if you had enough antibody and if the virus that infected you was susceptible to the antibody, that you were protected. And this was done by using a sieve analysis on the breakthrough infections, which showed that only in the control group were there infections with viruses that were susceptible to the serum levels that were achieved at VRC01. And the implication of this is that monoclonal antibodies could work, but you would require a combination of antibodies that target multiple neutralizing epitopes on the viruses that are actually in circulation. And of course, these should be non-competing so that you can have multiple targets, have high breadth and potency, and ideally formulated for sub-Q delivery. The AMP trial demonstrated that it is feasible to give IV monoclonal antibodies, but that route would preclude a large number of individuals in low and middle income settings from actually being able to receive these because of the medical facility requirements that are necessary to deliver IV antibodies. And more recently, there's been modeling data that suggests that a combination of 3 bNAbs should be sufficient to protect against most circulating HIV viruses. Now, the challenge for global access is that the most optimistic current cost of goods for CHO-based monoclonal antibodies are in the range of $50 to $100 per gram. And at the doses that were used in the AMP trial, a triple monoclonal antibody cocktail may be technically feasible, but the cost would be prohibitive for LMIC. And therefore, IAVI's focus has been on developing highly potent, long-duration bNAbs. And just as an example, this is a graph that shows on the x-axis the geometric mean IC50 in micrograms per mil of a range of antibodies that have been produced by a number of investigators. And on the y-axis, the breadth, the percent coverage that these antibodies have against circulating viruses that have been, this is done in a pseudovirus assay, but it's thought to be representative of what's happening in the real life. And you can see that the VRC01 antibody is out to the right on the IC50. It's got good breadth, but it's not very potent. And using a variety of methodologies and within the IAVI Scripps collaboration, we have worked on looking at how to, first of all, how to identify parental antibodies, and these are coming from individuals that have been, are categorized as long-term non-progressors that have these antibodies. These are rare individuals, but these are naturally occurring human monoclonal antibodies that can be engineered either through what's called directed evolution, using phage display and mutating essentially every single antibody in the CDR regions of these antibodies to increase potency, or through what the VRC uses as a more structural approach. You can see that the antibodies that are shifted to the left on this graph without dropping their breadth are much more potent, at least 10 times, in some cases 100 times more potent than the VRC01 antibody. And these are the families of antibodies that we think are worth progressing as a combination product. And we're currently working on the E, that's an engineered PGT121, an engineered PGD 1400, and working with the VRC on VRC01.23LS, which obviously are all considerably more potent than the VRC01 antibody. And as I said, our principal focus has been increasing potency while not reducing the breadth of coverage. And just as an example, these are data from in vitro neutralization panels using a large panel of pseudoviruses done at Mike Siemens' laboratory. If you look all the way over on the left, you can see the neutralization IC80 for the VRC01 that was used in the AMP trial, and it's the geometric mean was somewhere around 9 micrograms per mil, but 5% coverage. And if you now go all the way over to the right and looking at the triple combination of the engineered antibodies, you can see that we have potentially 94% coverage in terms of breadth and potency that's well down below 0.0 micrograms per mil for an IC80. So this is the level of potency and breadth that we think could actually have an impact in the clinic against wild-type circulating viruses. In order to develop this, we've created a broad partnership that includes funding and support from USAID and PEPFAR. We have a strategic partnership with them. We're collaborating closely with the VRC, who are providing one of the antibodies, and of course have tremendous experience in developing and producing these antibodies. As you've heard from Bob's talk, we're working with the Serum Institute of India, who have great experience in low and high volume manufacturing for antibodies and vaccines, and have distribution and access channels in lower and middle income countries. Umesh Shaligram will be on the discussion panel representing IAVI, representing us Serum on this topic. We've been working with the Wellcome Trust, as you heard from Shelley, to have a global call to action for expanding access to monoclonal antibodies, and of course, working very closely with the WHO on HIV antibody guidance documents, and using this really broad collaborative approach. We think we have a pathway into the clinic that will hopefully get us into our phase 1 studies late next year. Now, in terms of our target product profile and our alignment with WHO, our indication is for prevention of HIV infection in confirmed HIV-negative individuals, neonates, and infants with HIV exposure. The target population are people substantially at risk and their sexual partners. Access and affordability is absolutely key and underpins everything that we're doing in our development program. The efficacy has to be substantial, recognizing that these populations also have access to effective antiretroviral therapy. And so we have to certainly build a use case in the context of existing antiretroviral approaches. The formulation, we would prefer a single vial product. For infants, perhaps 0.5 ml dose. For children age 5 or younger, perhaps a 1 ml dose given subcutaneously. We may have to administer every 3 months, but obviously longer would be better. And all of the antibodies that we're working on are the LS extended half-life versions. Ideally, subcutaneous or intramuscular injection would be preferred for a commercialized product because of the simplicity of delivering these. For proof of concept studies, we may have to work with intravenous administration while we work on formulation challenges for combining high potency combination antibodies in the clinic. Looking forward to the future, there are obviously ways that we might be able to reduce costs of production in mammalian systems. These include facilities with smaller footprints, single-use reactors, perfusion cell culture, continuous processing, process intensification, integrated manufacturing, and potentially miniaturized mobile or benchtop bioreactors with modular manufacturing. We think this is feasible, but it's still some ways downstream. There are downstream process improvements, purification by crystallization or chelation steps, and non-traditional, in other words, non-protein A chromatography resins, protein A being one of the major cost drivers for purification of monoclonals. Alternative expression systems, if we can overcome the issues of glycosylation the differences in glycosylation that yeast, fungus, and algae and plants potentially produce, but certainly can produce high yield, low cost production. And we're working with Moderna, and you're going to hear from Obadiah later on about mRNA approaches for delivery of monoclonal antibodies. And finally, packaging, formulation, and delivery devices, formulation approaches that minimize cold chain requirements, achieve high concentration for sub-Q injections, and delivery devices that enable alternative deliveries from conventional IV routes are all going to be important in reducing the overall cost of a program that involves multiple monoclonal antibodies. And finally, we have had some discussion about these bivalent and trivalent, bispecific, trispecific antibodies. Both kinds of antibodies for HIV have been developed and look promising, at least in vitro. I think at least one of these has shown efficacy in a macaque model. But we certainly can see with the use of these bispecific, the graph in the center of this slide shows the parentals in dashed lines, and to the left, shifted to the left, a significant increase in potency in the case of a bispecific antibody. So this would allow you to have that increased potency and have manufacturing only a single antibody or perhaps two antibodies to reduce cost of goods. There are other challenges in terms of immunogenicity, off-target binding, et cetera, that have to be investigated with these novel designs, but we think this is a promising area for the future. I'll stop there and again thank our donors.
Thank you very, very much, Rip. I know you're actually on another meeting today. Do you have time to take a couple of questions from the Q&A?
Sure.
Okay, so one of the questions is around which antibody platforms were used to discover the HIV neutralizing antibodies that you're using?
Well, the approach was to do seroepidemiological studies where we collected B cells from individuals, large numbers of individuals, and these were done with our collaborators through the advanced program mostly in Eastern Africa. And essentially B cell sorting and screening these antibodies for potency, and then the engineering part of this for the work that was done by Devin Sock and his group at Scripps has been this so-called directed mutagenesis of through the CDR regions for selection of high potency. They're being expressed in CHO-K1 cell lines, so I hope that answers the question.
Yeah, no, I think it does. Thank you. And then there's another question about which are the main challenges for subcutaneous and intramuscular formulations, and maybe this is with regards to just the sheer volume that you can only inject so much. So for high volume products, and especially where there's 3 antibodies, I'm guessing that's what that question is
right about. The potency for for example, the VRC01 antibody was given at 10 and 30 mg per kilogram to adults. That volume just cannot be given without a lot of discomfort to an adult by a sub-Q route, which is why it was given IV. Our hope, if we can increase the potency 10 to 100-fold of these antibodies, we're now down to a volume of a few mils. And could that be, if that bioavailability remains in a sub-Q route, that this, in fact, now is something that is tolerable and feasible to deliver through a subcutaneous or intramuscular route.
Well, thank you so much, Rip. Great work that you guys are doing over there, and with all of your collaborators as well. So I'm looking forward to hearing from Moderna tomorrow, and also from Umesh about what Serum Institute of India is doing.
Great.
Thank you. Okay, and now I'll hand the floor over to Ruth Caron, who is at Johns Hopkins University, but she's also the co-chair of our Product Development for Vaccines Advisory Committee at WHO. And she probably knows more about RSV than anybody I know. So she's going to cover the long-acting Rsv monoclonal antibodies. Over to you, Ruth. Thank you.
Well, thank you so much, Erin, for the opportunity. And I have to first start out by saying that RSV and my presentation differ quite a bit from the two that you heard before, malaria and HIV. The first thing I have to say is I do know something about RSV, but I have not been involved in the development of these monoclonals. I have been an admirer from the sidelines, but I think, in fact, some of those developers are in the conference today and may want to provide additional comments. But the other important thing that I want to say that is really different from the two diseases that we just heard about is that the goal for RSV disease prevention, whether it's by delivery of vaccines or by delivery of monoclonal antibodies, is not actually sterilizing immunity. The goal is to prevent severe disease, which in the case of infants and young children is lower respiratory tract illness, hospitalization, and death. So just for those who are maybe a little bit less familiar with RSV, it is the leading cause of infant hospitalization with bronchiolitis and pneumonia. The estimate is that there are 1.4 million hospital admissions for ALRI in infants in the first 6 months of life each year, with close to 50,000 RSV attributable overall deaths. RSV has been found in every country in which it's been looked for, but as with many other pathogens, the greatest burden is in low and middle income countries, 95% of ALRI and 97% of deaths. RSV vaccine development was really hindered for many years, and I think most of the people listening will be familiar with the issue related to enhanced respiratory disease following administration of a formalin inactivated vaccine. We've come a long way. There are very promising vaccines in development, but there are additional issues related to protection of the young infant. And that really has to do with the infant immune response, the active immune response, and particularly suppression of that response by maternal antibody. So really the goal in terms of thinking about protection of the very youngest infants, there are different approaches to protecting older infants and toddlers. But for protection of the very youngest infants, there have really been two approaches. One is maternal immunization with RSV fusion protein, and the other is delivery of RSV monoclonal antibodies to infants at birth or shortly thereafter. So we do already have an RSV monoclonal antibody that's licensed. We actually, the history of passive prophylaxis against RSV actually goes all the way back to intravenous immunoglobulin, RSV IgIV, that was selected to have high titers of antibody to RSV. And that was the first passive prophylaxis. It was then followed by palivizumab, which is an anti-RSVF fusion protein monoclonal antibody. Palivizumab had an overall efficacy of 78% in reducing RSV-associated hospitalizations in preterm infants. It is used in high-income countries and very high-risk infants in the RSV season. It's not because it's not efficacious against RSV in older infants. It's really because of cost issues. Palivizumab costs thousands per season per patient. And in addition, palivizumab is not a long-acting monoclonal antibody, so it has to be given monthly, 5 or 6 monthly IM doses throughout the season. What I think is extraordinarily exciting is that we will very likely have new products, new RSV longer-acting monoclonal antibodies licensed for use within the next couple of years. This is, I think, a remarkable moment. These are monoclonal antibodies that have extended half-lives, a different mutation than you've heard about in the previous two talks. This is the YTE mutation. A single IM dose protects for at least 5 months. And I would say that this is really, this will become the standard, I think, for RSV prophylaxis in the future for young infants. So I think we will see the disappearance of palivizumab and of use of these monthly injections in favor of these longer-acting monoclonal antibodies. And indeed, the RSV monoclonal antibody PPC that Aaron mentioned at the beginning specifically talks about long-acting monoclonal antibodies. They're produced on CHO cells using newer manufacturing technologies, which makes them more affordable than palivizumab. And so the estimated cost of goods for a 50 milligram dose could be less than $5 US. So just to mention, and what you can see on On the right-hand side of this slide is a cartoon of the RSV fusion protein with the various antigenic sites. These were originally mapped by Jason McClellan and Barney Graham at the VRC. And this information has been hugely important in the development of these monoclonal antibodies. So the first of these to mention is nirsevimab MEDI8897. This is an antibody directed at the site 0, which is at the apex of the pre-fusion conformation of the RSV-F protein. This has been developed by AstraZeneca in collaboration with Sanofi Pasteur. This monoclonal antibody has been evaluated in phase 3 studies. It showed 74.5% efficacy against medically attended RSV-associated lower respiratory tract illness. It did meet the clinical endpoint in that study. It did not actually meet, although it was 62% efficacious against hospitalization. The confidence interval was wide, and it did not meet the pre-specified endpoint for hospitalization, most likely because of the perturbation of the RSV seasons by COVID-19, and really an issue, I think, primarily related to small numbers. When the investigators did a pooled analysis of efficacy against hospitalization that combined some of their data from a earlier phase 2B trial with the data from the phase 3 trial, they showed an overall efficacy of this product against hospitalization of about 77%. And this product is under review. I think we'll hear from stringent NRAs in 2023 about this product. A second product is also very far along in development. This is a product, MK1654 clesrovimab, developed by Merck. This is a product that targets site 4 on the, you can see it there, on the RSV fusion protein. The phase 2B, phase 3 trial is ongoing with expected completion in 2024. No data published to date on efficacy results, but moving along quite quickly. And then just also to mention that the Gates Medical Research Institute has also begun studies with a product called RSMO1, which targets, again, site 0 on the RSV-F protein. That's relatively early in development. Phase 1 trial is ongoing. So I think this is, as I said initially, I think this is really a remarkable moment in RSV monoclonal antibody development. I think the task at hand is really going to be to see how we can get these really very promising products into LMICs, where I think they really will do the most good. So there we go. Okay. RSV monoclonals for LMICs are approved under the current Gavi VIS, contingent on price. And modeling studies suggest that monoclonal antibodies could be cost-effective in LMICs under certain price assumptions. As I mentioned, how this moves forward, I think, really remains to be seen. Sorry. Okay. That's really all I had to say, and welcome questions if you have them. Thank you.
Thank you very much, Ruth. There was a question in the chat about whether or not there are other ways to deliver RSV monoclonals. And I do remember there was a product that had been developed by researchers at the University of Utrecht that looked at nasal administration of palivizumab. I don't know if you know anything about that. I haven't followed what's happened there, but I think maybe there was adherence issues with thinking about parents forgetting to do the daily nasal drops.
Yeah, I will say that I don't know about that study. I actually was involved in a study that was done more than a decade ago of actually an IgA monoclonal that was given intranasally to prevent RSV, and that particular product did not work.
Okay.
I think the other issue is really programmatic, and I think giving a single dose long-acting monoclonal, one time, we'll see if RSV seasons go back to something resembling to what we knew before the pandemic. Remains to be seen, but even so, having a product that will provide protection for 5 or 6 months, given as a single dose, even parenterally, I think will really be programmatically much more feasible than something that requires repeat administration, even if it's mucosal.
Yeah, and I think the nice thing about these products is that they can just slot so nicely into the routine immunization program, either as a birth dose or in one of the usual sort of routine immunization visits. But obviously the pricing of the products remains unknown for wide access at this stage. There is also a question in the chat about, in the Q&A about how well do these monoclonal antibodies protect against both A and B RSV? And is there any concern for viral escape?
So that's a wonderful question, and we actually, something that was not on the slides that we didn't talk about, but I'm very glad this was brought up. There was another RSV monoclonal antibody product that was developed by Regeneron. That monoclonal failed. And the specific reason it failed is because it did not protect against recent RSV-B strains. And you actually, when people did the sub-analyses of efficacy against RSV-A and RSV-B, they found, in fact, very good efficacy against A, but almost no protection against RSV-B. Both the Merck and the AstraZeneca Sanofi products that I mentioned have very high activity against RSV-A and RSV-B. I think, so that's current state of the art, and people have not yet detected escape mutants. I think that that's something that's going to have to be looked at continually as these products move into broader use.
Well, thank you very much, Ruth. So I'm presenting the next presentation, which is about RSV monoclonals. I'm going to remember which screen I have to share. Hopefully you can all see that, because I've lost view of it. Can everybody see my screen?
Yes, we can see.
Okay, fantastic. Thank you so much. So RSV monoclonal antibodies, this is quite an easy one because we've already got products on the market. But basically, just to cover a little bit of disease burden, is that even though we have vaccines and we've had rabies immunoglobulins for decades, 100 years or more, there's still approximately 600,000 deaths yearly, give or take a few thousand, due to rabies. And the majority of these deaths are due to dog bites. And most of these deaths are occurring in poor rural populations in Africa and Asia. An estimated about 15 million people around the world undergo post-exposure prophylaxis for rabies every year. And that could be in all countries of the world. But the majority of deaths are really occurring in Africa and Asia. So one of the WHO recommendations for severe rabies exposure, and this is people that have been bitten by a rabid dog, is to conduct wound washing, to administer rabies immunoglobulin directly in the wound. And then followed by subsequent several doses of vaccination at several days apart. But the reason that the rabies immunoglobulin is given is really to neutralize any residual virus that's left at the wound site after washing, because obviously that vaccine response takes a little bit of time. And as the rabies virus, sort of before it gets to the central nervous system, it's really important just to make sure, none of it actually passes through. If you've got a sort of superficial bite on a lower limb, RIG might not actually be needed. The chances that your active vaccine immune response will be sufficient is probably okay. But if you've got a more severe bite and the bite is closer to your neck and head, then having that RIG administered is really, really important. But the rabies immunoglobulin products in the past were made from either hyperimmunized horses, so equine rabies immunoglobulin, or from humans donors that had been hyperimmunized with rabies. And because of that sort of production technology, there were major issues with the supply of those products. And an estimated less than 10% of patients who were actually category 3 exposed, who should have required RIG, were actually getting it. They're just obviously human donors to, first of all, agree to give blood, but also to actually be hyperimmunized with rabies vaccine at a very specific schedule is a major limit to supply. HRIG costs thousands of hundreds of dollars, sometimes in the thousands. And then the equine products are obviously much cheaper than the human products, but it's a really lengthy process. I think it takes about 6 months to produce a single batch. And if there's any issue with those batches, they need to be destroyed, and this impacts supply. Horses need to rest in between campaigns. There's a whole schedule of, when they receive the vaccine and when you can bleed them. And it really is quite an archaic production technology. So back in 2002, WHO actually initiated a project to support the development of rabies monoclonal antibodies. We received donations from our rabies collaborating centres of hybridomas that were producing monoclonals that they had isolated. And we transferred those hybridomas off to some low and middle income country manufacturers for further development. Unfortunately, only one of them was actually successful in bringing a product to the market, due simply because of funding constraints with conducting the preclinical and clinical studies. But certainly for 20 years, WHO has been pushing for these products because they are so much easier to produce, even though we know that monoclonals are complex, but easier to produce than the blood-derived immunoglobulins. You can achieve mass production, and the quality control is, of course, easier. And the products are actually also more affordable than HRIG and cost comparative. Potentially, they could eventually be less than the equine rabies immunoglobulin. So in 2018, WHO actually recommended updated its recommendations around rabies post-exposure prophylaxis. And we now state that if available, the use of monoclonal antibody products instead of rabies immunoglobulin is encouraged. They're also included now in the WHO model of essential medicines list since last year. But at this point in time, there are only 3 producers, and they're still trying to gain their feet in the market. So for now, we need both the blood-derived rabies immunoglobulin and the monoclonal antibodies to ensure adequate supply at a global level. So I mentioned that there are 3 products that have been licensed. There are 2 producers in India, the Serum Institute of India, which I believe developed their product in collaboration with Mass Biologics in Boston. And then there's the Zydus Cadila product, which had received the donated antibodies from WHO. And there is a Chinese manufacturer that just licensed their product, I think, sort of in the last year or so. And we don't have a lot of information about that. But for the two Indian producers, the dose is actually so low because it's almost a topical application. It's administered at the site of infection, and the cost is very similar to the equine rabies immunoglobulin, which is really promising. So just to say, I think it's a lot of progress. It took 20 years, but we now have 3 products licensed, and there's also an additional product in phase 3 by Cinnamore. So far, at least the Indian producers are pricing their product at a very similar price to equine rabies immunoglobulin. One issue that we have at the moment is that there is no WHO pre-qualification pathway yet for rabies monoclonals. They're not approved by stringent regulatory authorities yet. So this is a barrier to getting a pre-qualification of those products set up. But hopefully in the future that might change. So that was it for rabies monoclonals. I'm going to check if there's any questions in the Q&A. And I don't see any with regards to rabies, but there are some with regards to malaria and RSV. So I would ask maybe if Bob and Ruth are still on the call, if you want to address those directly into the Q&A, that would be great. So I'm handing it over now to hear about another blood-derived immunoglobulin replacement. And that's for diphtheria. So Mark Klempner from Mass Biologics is going to talk about the candidate that they're developing to address the shortage in diphtheria antitoxin. So over to you, please, Mark.
Get this up. Erin, thank you very much. Can you see the presentation slides?
Yes, I can. Thanks, Mark.
Very good. First of all, let me start with thanks to the organization for inviting us. And let me just make a couple of comments on a couple of things that have been said already. First, I noted at the very outset your description of some of the currently licensed monoclonal antibodies. And I just want to give a shout out to Mass Biologics of UMass Chan Medical School, which is responsible for 2 of those. Bezlotoxumab, or Zimplava, and Rabishield were both invented here at Mass Biologics, and we've been at this for a long time, as you'll hear with my talk about EDAT and trying to replace it with a modern human monoclonal antibody. And the other thing, just on the rabies topic, I think it's important to note that the FDA has also provided guidance for the development of monoclonal antibodies for post-exposure prophylaxis. And they have unequivocally said that they will not entertain applications that do not contain at least a cocktail of 2 antibodies or more, because of the concerns of lyssaviruses and other that I saw in some of the questions there. So that product development pathway, at least for licensure in the United States, will require at least a cocktail of 2 antibodies. So now on to my own talk. It has faced different challenges than some of the others, being a real orphan product where we're trying to replace a currently licensed in some places product of an equine antitoxin. Currently, diphtheria antitoxin is part of the model list of essential medicines. It's been there for a very, very long time, and that is the current Indication for it is for the treatment, not prevention, but for the treatment of diphtheria. To remind people, diphtheria is largely a disease of displaced populations because it is a disease that is vaccine preventable in children largely. We have all been vaccinated with our TD or our Tdap as children, children across the globe are. It's caused by a bacterial infection, Corynebacteria diphtheriae, which is toxigenic. It produces a toxin that is quite inflammatory. It is absorbed into the bloodstream, where it actually involves the heart muscle in particular, and also causes great amounts of inflammation of the upper respiratory and lower respiratory tract. It has a mortality of something on the order of 15% in children and 10% in adults. And it is a disease that we see very little in the developed world, but continues to occur globally and actually has been increasing. I cite some WHO data here of most recently reported cases in 2018, where there were over 16,000 reported cases of diphtheria. As a result, WHO has also noted marked increases in the number of vaccinations that have occurred globally as a result of the COVID-19 pandemic. And we anticipate that there will be more cases of diphtheria, especially with global unrest. According to WHO, 23 million children missed out on their basic childhood vaccines in 2020 as a result of the pandemic. So there is clearly a need for the medicine. There's been a need for a long time in low-income countries. Mass biologics has really been part of the story of diphtheria treatment from its outset. Our very first product at Mass Biologics in 1894 was equine diphtheria antitoxin, and I sit in my office here about 100 yards from the stables in which this particular horse, Captain, was kept, where he donated his blood for about 80,000 treatment courses in the United States at the turn of the last century. Equine or EDAT has been the cornerstone of therapy since that time. It is a toxin neutralization monoclonal antibody, and along with antibiotics is the treatment of choice for diphtheria, to both kill the bacteria and to neutralize the toxin that it's producing. In addition to the need to replace an equine product, even the availability of EDAT has been tenuous over the past 20 years or so. And in many places where it was licensed, there was production stopped. There has been a very big move by some organizations to stop the use of horses entirely for the generation of equine hyperimmune sera. And all of this has led to an erratic supply. In many places, including the United States, until most recently, there was an outdated stock of EDAT. It has been renewed, but it is still an IND protocol-based medicine. It is not a licensed medicine in the United States, as it is in most other countries, and there continue to be relatively few sources of equine DAT. So our goal was to develop a human monoclonal antibody to neutralize diphtheria toxin and replace the equine diphtheria antitoxin. The reasons for this, of course, were to increase safety as we know, the safety profile of all of the hyperimmune antitoxins from or anti-enzymes from horses have a side effect profile of foreign serum products and very different than human monoclonal antibodies. And in addition, we were very, of course, focused on improving accessibility for this medicine that is really restricted almost in its use to places where diphtheria occurs and is difficult to reach and treat. We used a very standard, we've been at this for a long, old time. And so in this particular case, for our development of this monoclonal antibody, we used our own vaccine, a TB vaccine. We hyperimmunized some volunteers. We sorted B cells, collected and sorted B cells, screened them for antibody production and neutralization of diphtheria toxin, and then looked for the best clones that were both productive and making an antibody that was able to be downstream processed well. Some of the problems that I've heard alluded to in antibody production did occur with this antibody, including a major problem with aggregation, which was addressed with some germline changes to the amino acid structure of the antibody. Ultimately, though, we did select a high potency antibody. We call it S315, and this is the antibody about which I will talk about going forward. S315, this IgG1 human monoclonal antibody, was first, after tested in vitro, was first tested in the most appropriate model for diphtheria, and that is the guinea pig model. Because guinea pigs are highly susceptible to the toxic effects of diphtheria toxin, it does cause heart problems, and it also causes paralysis of their hind limbs as well as death. And you can monitor these both for symptoms as well as the protection from a lethal injection. When we did the initial studies, we did them with antibody and toxin mixed together. And it was, of course, very able to neutralize the toxin amount. And you can see on the left that doses above about 2 international units were highly protective of animals given a fixed dose of toxin. And that also turns out that above about 20 micrograms of 27 or 37, excuse me, micrograms of the monoclonal antibody, there was also full protection of animals when it was mixed together. Of course, that's not how patients present. They present after they've been intoxicated. And so to follow on this, we did the rescue experiments in animals to show that the monoclonal antibody protected both from symptoms and death. And you can see here that above 0.175 milligrams of S315, there was almost full protection of the animals for an intoxicating. And if you look at the bottom of this, at the 7 milligram dose, which had the largest number of animals in it, these animals were, again, 95% protected at that dose. That gave us encouragement to pursue it further with further preclinical studies that are necessary in the regulatory pathway. And we first looked at animal toxicity, and we did not see any clinically significant toxicology in all the blood studies that were done on those animals. We did tissue culture cross-reactivity with a standard human and rat tissue panel, of S315, and there were no toxicologic significant reactivity. And we did the typical 15-day rat toxicity with a 28-day recovery period. And the no observed adverse event level was greater than 500 milligrams per kilogram IV, which was the highest dose tested in the animals. So all of our preclinical studies pointed to a non-toxic, highly efficacious monoclonal antibody that we further developed in the next phase, which was our first in human phase 1 trial, which was completed 2 years ago. And you see here the objectives, which were safety, tolerability, and pharmacokinetics of S315 in normal, healthy volunteers that were adults, 18 to 55 years. There were sentinels receiving the placebo and sentinels receiving the S315 at each dose. These were IV infusions. Recall that diphtheria treatment is a single infusion treatment at the time of acute infection. So we're not worried about redosing. And of course, we're not necessarily worrying about route of administration. This is all delivered intravenously, as all acute therapeutics would be in this case. We did observe all of these volunteers for 60 days for toxicity, and you can see that the comparable dose to the recommended diphtheria treatment for diphtheria of 80,000 international units was about 3.8 grams of S315. You'll see shortly that that is probably a very large overshoot based on the potency of the antibody, but that's what was delivered to people. And in summary of the S315 phase 1 study, there were basically no adverse events that were attributable to the S315. There was one suicide in one of the volunteers, but was deemed unrelated to the infusion of S315. The other adverse event profile of S315 infusions in these normal volunteers really favored the monoclonal over the placebo for at least any AE, and there were no SAEs, as I said. The most frequently reported AEs were in the placebo group of headache and upper respiratory tract infection occurred in 3 of the 30 people who got S315, again, thought not to be related. It was obviously a very safe monoclonal antibody. Of great importance is the pharmacokinetics of S315 because we are trying to neutralize a toxin dose that is present at the time of infusion. And for comparison, when we studied the neutralizing capacity of EDAT, and this is a published paper by Smith et al. In 2017 from infusions of EDAT, the mean concentration of neutralizing capacity in a virocell assay of an EDAT infusion of 80,000 international units was 34.6 neutralizing units per mil. If you look now to the right here of the various doses of S315 that were administered, the comparable 80,000 international unit dose of 3,840 milligrams delivers 722 neutralizing units per mil in the virocell assay, roughly an order of magnitude greater. So we think that this dose is greatly overshooting, but it would be the starting dose, I would imagine, in a field trial. We have a lot more about the pharmacokinetics here. We're not worried about long-term duration, like with the LS mutations and the YTE mutations, et cetera, because this is an acute toxic intoxication, and we're trying to eliminate that. We've been in touch with the US FDA and with the results of our phase 1 trial. And as you know, in the United States, there is no licensed DAT. The available product is an unlicensed compassionate use product, which is EDAT. The US FDA has approved the use of S315 for compassionate use in the United States by the CDC, and the CDC is in the process of filing its IND, investigational new drug application, and it will reference ours, and we are doing this in collaboration with them. They then will be in the position to stockpile S315, and it will be available through compassionate use. We've also been in touch with the FDA in navigating the future regulatory pathway. And as you can imagine, doing a field trial for acute diphtheria cases to prove efficacy, which was, by the way, never done with EDAT, would be very challenging. And so in consultation with the FDA, we have been advised to pursue licensure in the United States through the animal rule, which is a good laboratory practices study to demonstrate efficacy of S315 in the guinea pig model, which we're quite confident we will be doing. We will commence the required animal studies for licensure next year. And we anticipate that following those studies, which will take about 13 to 14 months, that we anticipate a BLA filing, a biologics licensing application in the US in 2024. I think that's occupied my time, and I really appreciate the attention, and I'll look forward to any questions. Thanks very much.
Thank you so much, Mark. I think you're right. I think we are going to see increased diphtheria outbreaks occurring. The slip in immunization coverage, has been quite, well, maybe not surprising, but quite shocking. And certainly in displaced populations, refugee camps, this is where we're probably going to see these outbreaks occurring. And I think it is really fantastic that the US FDA has agreed to a licensure pathway based on the animal model, because I'm assuming through compassionate use only in the US, there's only so many cases of diphtheria in the US setting. So the demonstrating efficacy in humans is going to be so challenging. So that's really great news. And because it will hopefully be licensed by US FDA, that is considered a stringent regulatory authority. And that makes the WHO pre-qualification process much easier, which then would help with global access. So great work there. I'm just going to check the Q&A to see if there's anything specific for diphtheria monoclonals. And there isn't. So thank you very much, Mark. I'm going to hand the floor over now to Christopher DaCosta from CEPI. He's going to talk a little bit more about what the role of monoclonal antibodies might be in the prevention during or post-exposure prophylaxis during epidemics and pandemics. Over to you, please, Chris.
Thanks, and I'll share my screen.
You should have a green button at the bottom.
Yes.
Oh, yeah, got it. Great.
Yeah, can you see my screen?
Yes, we can. It's not in presentation mode yet.
Yeah, it's just about going there slowly as we speak. Can you see that?
Yes.
Can you see my screen?
Yes, we can.
Yes. So basically, I was just going to address the use of monoclonal antibodies for prevention of diseases with pandemic potential, really from the CEPI strategy point of view. As you know, we're primarily a funding organization and also provide technical support for development of these products that are made available for widespread use globally, particularly in the context of poor or low resource settings like LMICs. So the focus is really going to be less technical than you've heard from other talks and more on the strategy. But there is a background to this. The public health need, which has been articulated before by several, is the potential role of these monoclonal antibodies to be rapidly deployable as countermeasures to either complement or precede vaccines in instances of outbreaks, be they epidemics or pandemics, globally, but particularly in low resource settings in this instance. In high income countries, the interest for complementary use of monoclonals has really been escalating since the onset of the COVID-19 pandemic, as we know, and I'll show a slide later on to go into that a little bit more. The utility of these monoclonals is also been looked at as a potential for protecting those individuals that may not respond to vaccines or that have particular vulnerabilities and require more rapid introduction of some form of treatment or prophylaxis. There are several benefits of monoclonals that also have a knock-on effect in reducing the reservoirs of infection and limiting transmission that also have been noted. And these are also part of the overall public health need that needs to be further explored.
Sorry to interrupt, Chris. We actually do see your slides, but it's in the view, I don't know what you call it, but where you see the slide and then you see the next slide. This happens to me quite often when you have multiple screens.
Christopher, can you hear me?
Yes, I can.
Do you see the display settings mode at the top, above your window? Yes. Give it a try again.
You want me to stop sharing and then to share again?
No, that's fine. That's fine. Go, try to make it full screen again, please.
Full screen again. Is that better?
It's not showing yet. It's thinking. Yeah, sorry about that, because the presentation is small, and I think some participants struggle to see.
I actually have two screens here, and I see this.
So maybe stop sharing.
Yep.
I will guide you. So what you can do is before you share, can you have your PowerPoint stop sharing first?
Yeah.
Because you can still see.
Okay, let me stop sharing. Okay, share. Stop share.
Okay, great. So now what you can do is don't share yet. Have your PowerPoint in full screen, in presentation mode. Let me know when it's done. So if you have to,
yes.
If you have two screens, that's the best way. Now, what you should do is when you click on share screen, there's a little arrow. You will see several windows. Choose the window of the presentation in presentation mode. Let me know if you struggle or if you don't see it.
I don't exactly get it.
All right, so I will jump ahead. You were just finishing this slide.
Yes, and I think I was on the next slide already.
There we go.
Yeah, and so this slide really goes into the state of development of some of the priority pathogen monoclonals that CEPI is interested in, the first four. I've also thrown in Ebola just to illustrate that although most of these monoclonals are really at the point of having demonstrated preclinical proof of concept, Ebola is a bit further along. So for Nipah, we know that there is a neutralizing humanized monoclonal that targets the G protein that's cross-reactive to both Nipah virus and Hendra virus. I've put the publication in brief, the author and the year. and a humanized, another also humanized cross-reactive anti-F glycoprotein monoclonal that also targets both viruses. These are thought to be potentially prime time in terms of translation into the clinic. For Lassa, there is a cocktail of human monoclonal antibodies developed targeting both the GP1 and GP2 subunits that are cross-reactive with all 4 clades of Lassa. For Chikungunya, there also is a monoclonal antibody targeting the virus. More details in the publication. Not very sure how far gone that is in terms of prime time. And in MERS, there is a cocktail of 2 monoclonals that bind to non-competing sites on spike that neutralize the virus by blocking the DPP4 receptor and has demonstrated some clinical efficacy, some efficacy in animal models, a humanized mouse model. There also is data. There are data from a phase 1 trial of the cocktail in which co-administration of monoclonal antibodies was shown to be well tolerated. That's more recent data. So those have all demonstrated some preclinical proof of concept. For Ebola, as we well know, that a cocktail of monoclonals and an additional second monoclonal have actually been used in the field and demonstrated some degree of efficacy in the field. So for this is just really a preamble as to where CEPI is going with these products, supporting development of these products. On the next slide, I just thought I'd just delve a little bit into what most of us here will know, particularly as the use of monoclonal antibodies, mostly for therapy in COVID-19, but also in some instances approved for post and or pre-exposure prophylaxis, has sort of been a precedent for what might be in future for some of these priority pathogens and other infectious diseases. And so links that I've provided there mostly go back to the original WHO recommendations, but also recommendations from other entities. As we know, this is really a field that's changing very much. You know, the continued study of the target populations, the ideal target population spectrum of disease within COVID, for which there is ideal use of these products, continues to evolve. So we know, for instance, that the cocktails of casirivimab and imdevimab and tixagevimab and silgavimab well established as use cases for treatment. We also know tocilizumab established for treatment. We know that sotrovimab has had more recent pullback from original EUA for outpatient use. And that's for preventing hospitalization. But no longer authorized in the US because of its lack of ability to neutralize some of the more recent subvariants of Omicron. So the field's changing. I think the key message here is that although these were used primarily for treatment, that some of these, especially the cocktails, do have a role in post-exposure prophylaxis and in pre-exposure prophylaxis potentially as well. I think the important thing in terms of CEPI's policy or CEPI's vision is about the concept of 100 days, which many of you know about, during which there is a time frame in which, albeit a shortened time frame to developing vaccines that might be deployable with a very short target, there is still a time period between onset of an incipient epidemic or pandemic and the actual institution of vaccine efficacy after you deploy vaccines, during which there could still be significant mortality or morbidity from an outbreak. And this is where some of these monoclonals, if they are developed for off-the-shelf use, can be used in high-risk groups for pre-exposure prophylaxis and more widely for post-exposure prophylaxis. On the next slide, just a few of the critical challenges. I'm not going to belabor this because these have been articulated multiple times in previous presentations. We've talked about high cost of goods, in part related to potency issues, the requirement for intravenous administration of existing monoclonals, with the exception that some of the cocktails that I mentioned on the previous slide can be now administered either intramuscularly or subcutaneously in addition to IV. There was a mention in one of the previous presentations about the utility of IV administrations for relatively short periods of time that could still be used, but always a concern in LMICs for some of the logistic reasons that were mentioned before. Cold chain requirements and requirements for, not just for transportation, but also long-term storage and stockpiling are critical for these products. And also a whole myriad of logistic problems related to deployment in a cost-effective manner in these resource-poor settings. So a lot of challenges in actually getting these products for more widespread use in these countries. The next slide goes into a little bit of some of the ongoing efforts. Again, some of this has been said before. I'm not going to talk too much about this slide because I know my colleagues from the Gates Foundation, I know that Jacqueline Kirshner tomorrow will be talking a lot more about biologic therapies, and we'll go into more details about this. But some of the approaches are to optimize existing monoclonals, to introduce those technological innovations that would increase potency, as we heard of before, and allow for alternative routes of administration to be more widely used, and therefore make them more amenable to use in LMICs, to develop novel manufacturing platforms, and also novel delivery methods, such as gene delivery methods, including mRNA. The alternative biologic therapies, again, as I said, I won't go into details from this slide, but the DARPins and nanobodies have generated a lot of interest, including, in addition to others, which I'm sure Jacqueline will talk about tomorrow. And it's very important that these products, when developed, will pay considerable attention to matching the product characteristics to the target pathogen, and very importantly, the use case, whether this is PEP or PrEP or therapeutics, given the differences, particularly in half-life of these antibody-like products compared to full-size antibodies that may be used in this context. On the next slide, we go more into the specifics of what CEPI is doing from a strategic standpoint. This is an ongoing effort with a therapeutics task force that's been developed at CEPI to address this. I also have here, actually attending this meeting, some of my colleagues from the technology group who are very instrumental in helping to craft this policy, and who you'll be hearing more from in future, I'm sure, in these talks. Stacey Wooden and Rebecca Farkas, I believe, are both here. And the monoclonal antibodies, as we know, have becoming more and more recognized as a critical tool that could be useful in outbreak control. We have very good examples, tangible examples now with Ebola and with COVID-19. And CEPI's plan is really to look at PEP and PrEP as the primary goal in use cases for select pathogens where they may be considered to be a better alternative to vaccines or complementary to vaccines, as I mentioned with that 100-day concept. CEPI is not going to be involved in de novo development of products, but rather taking those products that have, at a minimum, established preclinical proof of concept, and then help to translate these into the clinic to support them both with funding and for technical support, for translating these products into the clinic. There is a clear recognition that this should be a collaborative effort, that there are various academic, biotech, and industry groups that are working on this. These include NIH, NIAID, and CEPI is working closely with several groups to support this. In particular, we have a very strong collaborative effort ongoing with the Wellcome Trust, and I think Lindsay and Pete, both here, from the Wellcome Trust, and also my colleagues I mentioned before, Jacqueline and Steve at the Gates Foundation. So we work very closely in developing CEPI's policy moving forwards. The primary focus, the policy is nascent, but we know that the primary focus will be for monoclonal antibodies for post-exposure prophylaxis in those high-risk groups for exposure in healthcare settings, for NEPA, and also for pre-exposure prophylaxis where this is deemed necessary. So NEPA is a priority, as the #1 priority for CEPI. We expect that sometime in 2023, there will be a call for proposals for translation of NEPA antibodies that have monoclonals that have developed a level of preclinical proof of concept into the clinic, and support to fund further advanced clinical development and downstream efforts to license these for further use, for future use. The anti-Nipah monoclonal antibodies are thought to have a potential to fulfill a critical gap prior to vaccine availability, prior to induction of protective immunity, as I mentioned in the 100-day concept, for example. Beyond Nipah, CEPI does have tentative plans to look at Lassa potentially, and even MERS as targets for this kind of therapy. But as I mentioned in the previous slide, there are many other pathogens, and others have mentioned pathogens I didn't list that also engaging in the use of monoclonal antibodies for prophylaxis or for therapy. And so that's just a general overview of what we're doing at CEPI. As I said, I think in future you will hear more from us about things as things advance more. I just finally wanted to acknowledge some of my colleagues who've been working very hard on this, both in the technology office, Becca and Stacy, who are both here, and also some of my other colleagues in the therapeutics task force, as well as collaborators, as I mentioned before, from the Gates Foundation and from the Wellcome Trust. And I think I'll stop at that point in case there are any questions. Thank you.
Thanks very much, Chris. I don't see any specific questions in the Q&A. We've got about 15 minutes left. I see there's a question about rabies pre-qualification. I'll provide that question in writing. But I will I'll keep projecting my screen. I've got a Mentimeter discussion poll, and you'll see in the chat that there's a BIT link. If you could all copy that BIT link into one of your web browsers, it should take you to a Mentimeter poll where you can answer a few questions. And I'm hoping that you can see the poll now that I'm displaying on your screen. Christopher, since I can see you, can you see that Mentimeter poll? Okay, yes. So it looks like people are using it, so it's working. So you'll need to copy that BIT link into a web browser in order to answer the questions, and they'll appear in live time. So I'll give everybody about 2 minutes to answer that first question, and then I'll change to the next question. So do you think it will be viable and cost effective to use monoclonal antibodies to prevent infectious diseases? And it's looking like most people are saying it depends. And I think that that's very true, that it does depend on the pathogen and probably the target population as well. Obviously, for infants, a small dose can be achieved, which means cost of goods can be kept lower, which means that you can also have lower volumes, and intramuscular or subcutaneous administration is easy. All right, so there's definitely some consensus there on that it depends. Okay, I'm going to move to the next question. I'll just see there's still a few more coming through. All right, I'll move to the next question now. So for which pathogens would the development of monoclonal antibodies for prevention address an unmet public health need? So you can enter as many pathogens as you like. There's up to 10 pathogens per person. So you should be seeing that on your screens as well. And again, I'm just monitoring the time. I'll give everybody a couple of minutes to answer that one. Right, so it's looking like malaria, HIV, RSV, rabies, Nipah, influenza, Lassa, and Chikungunya are the predominantly answered pathogens, and then there's a lot of other ones that people are thinking that they could be used for monoclonal antibodies. All right, I'm going to move on to the next question. So in thinking about which attributes are needed to facilitate the use of monoclonal antibodies to prevent infectious diseases in low and middle income countries, which statements do you agree with? And this is from strongly disagree to strongly agree scale. So products should be administered via intramuscular or subcutaneous injection. So whether you strongly disagree with that statement or agree with that statement, you just have to move the dot across. Is IV administration acceptable? Strongly disagree or strongly agree. Products must be priced similar to vaccines. Strongly disagree or strongly agree. Products will be more expensive than vaccines. Strongly disagree or strongly agree, or somewhere in the middle. Cold chain requirements should be similar to vaccines. Strongly agree or strongly disagree. And products should be able to be stored outside of refrigeration for periods of time. So it's looking like, yeah, most people are saying that products should be administered via intramuscular or subcutaneous injection. Some of you think that IV administration is acceptable. And I think certainly in some settings, especially for hospitalized patients, and if you're using it as a treatment, I think that does make it much more feasible. Products should be priced similar to vaccines, and products will be more expensive than vaccines. Interesting that that's quite similar responses there, and quite similar responses for the cold chain requirements. I'll just give that another minute before I close the poll. Okay, and then the final question is, would the development of generic preferred product characteristics be possible and useful, or are these very much pathogen specific? Do we need to be seeing separate preferred product characteristics developed rather than having something more generic? All right, so it is looking like more of you think that these are pathogen specific, but there is a number of you that would see value in generic preferred product characteristics, and maybe that could cover certain attributes. Okay, well, with that, I'm going to stop sharing my screen and close the poll. Thank you to everybody for your time today. There are some questions in the Q&A that I'll attempt to address by writing. Tomorrow, we've got, again, a packed agenda. We're going to really hear from developers of some of these innovative technologies that might lower the costs of goods or increase the potency of the monoclonal antibodies. So some of the improvements that may lead to more affordable products. And then we're going to hear also from the FDA about some of the experience that they have. We'll hear from Bill and Melinda Gates Foundation as well about what they're doing to advance monoclonal antibodies for global health. And then we'll finish off tomorrow's session with a panel discussion. So hopefully see you all again tomorrow, and thank you for joining this afternoon session, or this evening, or this morning, wherever you are. Thanks very much.
Thank you. Bye.